Let's have a look at a scenario when you have the protein sequences available, but there's not a structure available for the protein sequence or not deposited in the protein data bank. And on top of that, you don't find an efficient sequence identity, let's say sequence identity over 30% with the template structures if you do a blast search for that matter with the target sequence. So this is a special case, and none of the previous methods could be applied in this case. So we already saw two different scenarios. The first one is basically you have a protein structure available in the protein data bank, and it's solved by X-ray crystallography. But there are certain regions missing in the structure, or there are certain residues which are not being able to resolve the structure. And then you can model them as loop regions by using the builder tool in PyMod. And the second scenario is when you don't have a protein structure available, but you have the sequence, the primary sequence of the amino acid residues available for that protein structure. And you model that protein structure based on some template structures which are homologous with that of the target sequence that you're interested in. And that homology should be at least 30% identical. Now, this is a third scenario where you don't have both. You don't have a protein structure as well as you don't have the homology with the known templates. So you resolve to methods which involves ab initio prediction of the tertiary structure or the tertiary folds in the protein structure from scratch uh, without using the information on the sequences. So this is basically structure-based modeling. And there are a number of ab initio methods that are available, but the one which we are going to focus on is called threading, and it's a very efficient web interface that you have. It's called iTasser by the Sang Lab in University of Michigan, which does the threading for you. So basically what you do is you divide your sequence that you obtain, and you're interested in obtaining a predicted protein structure from that sequence into different threads or different parts. And for each one of them, you try to get a hit template, that is, try to get a structural component for those fragments, and then finally join them together or stitch them together by different algorithms or different informations. It also involves some amount of artificial intelligence uh, in the process. And then you obtain the final structure of the protein. So it's a very de novo and ab initio kind of method. So simply put, threading is a fold recognition method to model proteins that have the same fold as the protein of known structure, but they do not have the homologous proteins with the known structure. So the model system that we'll be using for doing this threading with iTasser. But LAG3 has got a sequence homology of around 20 to 25% with that of CD4 and IL-2, which are the immune responders in the body. So it's responsible for T cell immunology. And these are also glycoproteins, and they have got a specific topography or the morphology in their structure with specific domains, especially four domains, four extracellular domains, which are separated by loop regions. So we are expecting that lag3 would also have a similar structure of that matter. But we can't do the homology modeling because the sequence identity is less than 30%. So the first thing that we need to do, and as always, since we are doing the prediction from the sequence and scratch, we'll have to look for the sequence in the UniProt KB database again. So we search for lag3 and followed by human. And this brings us to a page again where we find all the proteins that are listed with the hits of lag3. And the very first option here, the very first entry that you see here is corresponding to the lag3 protein. for the Homo sapiens, that's human, and it has a length of 525 amino acids. So it's quite big. So we go to this entry and we look for the sequence basically. Once again, you have a whole bunch of information on the UniProt page here for LAC3. If you scroll down further and look for the sequence, so you don't find a structural annotation for this protein. We go to the sequence and we look for the isoform 1 of the sequence, and basically the FASTA sequence is available, and you select the whole sequence and copy it to be used later as iTASSER input. Open a new tab and go to the iTASSER page. So type in iTASSER and it will lead you to the first hit, which is the iTASSER web page for doing the threading calculations. So you see the home page. So the server completed prediction of this many number of proteins submitted by this many number of users in 150 countries or regions. So it's extensively used by researchers across the globe. And it recently also solved the structural models for the coronavirus, which is fascinating. Okay, so this is an online server, and what you need to do is you need to have an email ID and password for yourself. So you need to register yourself first. I've given you the registration link already in the slides, and so you can go ahead and register with the UL ID and set a password for yourself. So every time you get to use this service, you'll have to input your email ID and the password in these fields. So we have copied already the sequence for LAC3. Now we are just going to paste it here. So just checking whether the sequence is fine. Yes, it is. FASTA format. And you can give your ID as well, optionally. So let's say the name of the protein. So I give LAC3 here. And you have additional options. There is option 1 for assignment of restraints and specific templates. So if you want to bias towards certain template, you can do that. But we are not interested in that. We'll let it do it in a free will, like we'll let it go without bias, search the database and everything. And you have option 2 as well. So if you want to exclude some templates, again, a different kind of bias. And the third option is if you want to specify the secondary structure for specific residues. Well, we don't need to do that because we don't have that information. We'll let the program decide for itself where the secondary structure regions might lie, which is the first step anyway in the structural prediction by iTASSER. So there's another option, keep my results public. That is, you have to uncheck this box if you want to keep your job private, which means if you're looking at something very confidential, this regard and a very novel protein. So we'll basically keep this result as private. So we uncheck this option. So once we're happy with it, we'll press on Run iTASSER. And the result link will be emailed to you from the server itself once the job is complete. So you can see I've received an email saying iTASSER results for LAC3, and it's from the Yang Zhang lab at University of Michigan, and says your job with job ID this has been completed on the iTASSER server, and the full prediction and everything is available at this link here. As if you try to access your job from the iTASSER Q page, you can also use the job ID and the results will be kept. So the results are kept in the server for 2 months here. So we're just gonna copy this link here and just gonna use a new web browser to open it. So if we paste this link here, and it will lead us to the iTASSER results for job ID, this here. So let's go through the information step by step that's given in the results page. So first of all, you get a compressed directory of the results as a whole, like all the information that's given out here. And you can download it directly by clicking on this link. So the first information that you have here is the input sequence. So submitted sequence in FASTA format, and it's automatically recognized as a protein sequence because of the amino acid 1-letter code that it has. The second information and the very primary step in the iTASSER prediction method is the prediction of the secondary structural features. So it has its own algorithm that closely relates with the dictionary of secondary structure for proteins, that's DSSP. And so it has this color coding for different secondary structure regions, which is red for helix, blue for strand, So two of the strands together forming a hydrogen bond would give rise to a sheet, and then black for coil. So as you can see, most of the structure here is either strand or a coil with a small region of helix that's formed. Well, it's not really a proper helix. It could be a 310 helix or a short helix that's formed around the middle of the structure, somewhere around here. So there's more to this. If you scroll down further, then you'll find another short region of helix out here. And then further down, there are pieces of helices, but most of the structure is either strand or a coil. Okay. So next we go to the predicted solvent accessibility. So the solvent accessible surface area generally gives you an idea of the amount of buried residues or the amount of highly exposed residues. So the residues which should be lying at the periphery of the structure, or the residues which should occupy the inside of the structure. And most of the buried residues should constitute the binding pocket to the ligands or the antibodies that are supposed to bind to LAG3. So the values range from 0 to 9, with 0 being completely buried residue and 9 being highly exposed residue. So we see here some of the residues out here in the N-terminal region are actually highly buried. That constitutes the D1 domain of LAG3. And if we go further down, it's a mixture of buried and exposed regions. So the lower the value, let's say the values lower than 5 would be more buried in nature, the residues, whereas the values that are higher than 5 would be more exposed in that regard. Okay, so towards the end, as we reach towards the C terminal, you also find, which is basically the D4 domain, and the last bit here is the intracellular domain. So this part is basically buried inside as well. And for that matter, none of the residues actually hit 9. So we can say that not much of the residues exposed in nature. So the more the exposed the residues, the more the propensity of them forming a random coil structure. So it's a very structured structure, to put it that way. That is, it has a lot of tertiary structural features having secondary structure folds. Okay. And then the next feature that we have here is the predicted normalized B factor. So you already know that the B factor and the temperature factor are numbers which give you an idea of the amount of flexibility in the structure. And they have this predicted B factor values in the form of a graph. And as you can see, the normalized values are having this standard value around here. And the structure, different regions, the different residues in the structure fluctuates around that region. So it's not very, very exposed again. If it was too exposed, or let's say if the flexibility was much more, or the B factor was very high, then it would have fluctuated to a much higher level, somewhere around this range out here. Whereas for the terminals, you see there's always a high fluctuation because they are completely exposed to the solvent. And they have the specific annotations with the secondary structure as well. And as you can see, most of them is strand and coil with a little bit of like flurry of alpha helices in between. If we scroll down further, we'll get the list of the threading templates that were used, which are basically the structures that were used as threads or small fragments to match up with the fragments of the sequences that we had submitted as an input. So it gives you a list of top 10 threading templates that were used by ITASSER to build the structure from our sequence. And it uses the database of LOMETS, which is coordinated with the PDB library. So it's a special kind of meta server threading approach that's been taken into consideration. And these are the PDB hits that you get with the PDB IDs and the corresponding chain IDs against it. So these are the 3 chain A's and then chain K and then chain A. So the first 10 hits, and you can download the alignment with our predicted protein structure as well with these templates. And the secondary structural features for these hits are also given out here, which should ideally closely correlate with the predicted structure from our sequence. If you scroll down further, finally, it will give you the top 5 models that it predicted from structure. And right away, you can see that this structure has close correlation with the CD4 or the IL-1 structure. That's the immune response proteins that are responsible for, and they are glycoproteins as well. And we'll see that in a minute. We'll look at the structure of CD4 and see how closely it correlates with this structure out here. So ideally, you would opt for model 1 out here. As you can see, when you go from model 1 to model 2, model 1 and model 2 are very similar to each other, whereas when you go to model 3 and model 4, they become slightly different in the structural orientation. Well, it's more compressed or globular in nature, as opposed to these ones, which are more elongated. And as we have kind of predicted that these would contain small chunks of domains of tertiary structure separated by small loop regions, and each of these would constitute domain 1 to 5, 1, 2, 3, 4, 5. There's an extra domain out here, which ITASSER has predicted for some reason, but we can always evaluate and cross-validate that with the known structures that we are anticipating to get the hit against. Okay. And once again, do not confuse this with homology modeling, because homology modeling is totally sequence-based approach. Although I keep on telling about the fact that we should always compare the two structures together, but I mean that from only a structural perspective, only and only structural perspective. It's nothing to do about the sequence. This has got like 30%, less than 30% identity with the template sequences. So scrolling down further, we'll get an idea about the proteins which are structurally close to the target in the PDB. So the target sequence that we had and the target structure that was generated from the sequence, and the alignment, the close alignment with that of the template structures, we'll get those PDB hits as well out here with the corresponding RMST and TM score. So TM align is a special program which does only the structural alignment as opposed to sequence alignment. And we get the structural identity here, the covariance, and the alignment here as well. So just to bring into your notice another thing which is very important is the normalized Z-score. So when predicting the threading templates, the normalized Z-score is a good metric in order to do those predictions. So the more the Z-score, the better the chances of getting a hit. Now let's get the last bit of information about the functional annotations that it predicts from the structural information or structural alignment that it generates. So the predicted function using cofactor and coach, so two different approaches combined together to predict the functional features of this protein, and also it does some prediction based on the ligand binding sites. So you see the prediction on the ligand binding site is given out here. So if you want to scroll on it and zoom inside, you will get an idea about the ligand names and the possible ligands that it could bind. Well, this doesn't need to be actually perfect in that regard, but the site or the predicted regions are usually of some value or of some information based on which you can design your inhibitors or antibodies or specific ligands that might be used to activate this receptor. Scrolling down further, you have the information on the enzyme commission number. So if it's a kind of enzyme, then it will predict for you the confidence score of the EC number prediction. And then you have the gene ontology terms, which are also functional annotations to a certain extent. So it's a treasure trove of information that you have out here. It finally gives you another link to download the file from here. So what we'll do is we'll go ahead and download the best predicted structure, which is this guy. So download model 1, you click on it. It will basically ask you to download it in your respective directory on your local machine. So you give model 1 lag 3 predicted as the name and give it an extension .pdf. So it will save the structure. So let's compare with the proposed known structure that we were anticipating that it would correlate with. That is the one which has got less than 30% sequence identity, but still, to a certain extent, the structural annotations uh, could be same to a certain extent. Maybe the domains and the regions might not exactly match up because they are not conserved in nature. The sequences are not homologous in nature, but the structural insights might be valuable. So I know the PDB code for the CD4 protein. So we go to the RCSB PDB homepage, and then we type in 1WIO, which is the PDB code for CD4, and it brings us to the page. It says structure of cell surface glycoprotein. So it's a glycoprotein as well, CD4, tetragonal crystal form. And right away you can see that there are four domains out here, which should ideally occupy the extracellular region for the glycoprotein. And if you go to the 3D view, you can get a better perspective of the structure. And so you can say that the structure actually closely relates to our lag-3 structure that we have predicted by using the threading method. So the accuracy and the efficiency of the method is proven. So you get a better view of the structure of CD4 by having a look at this. So these are the four domains: domain 1, domain 2, domain 3, domain 4. So let's open up our structure, the predicted one. So I'm opening that in Chimera directly from the download link that I have here, and just can place them side by side. Let's see. So there are four domains here, but in this case, actually, I've got 1, 2, 3, 4, 5, 6 domains. Ideally, there should have been 5 domains, but ITASSER predicts as a six domains. So the two domains together at the end could be considered as a single domain, and they should lie basically intracellularly in the intracellular region. Let's say. So this is the N-terminal domain, and this is the C-terminal domain, basically. Okay, so that's your ab initio structure prediction by the ITASSER web server.